The seventh leading cause of death in the United States is a virus that causes 1 billion cases of this disease yearly. This virus is called influenza, or more commonly, the flu. Although yearly flu shots are a common routine, it's important to understand the impacts of such a normalized disease in our society. The major building blocks of influenza include an 8-piece of viral RNA within the virus membrane. There are two main types of surface proteins surrounding the virus, called hemagglutinin and neuraminidase. These proteins have a head and stem structure in which there are many different variations in the structure of the heads of the proteins. Different hemagglutinin and neuraminidase protein structures cause different strains of the influenza virus, which are named after the combination of variations in the surface proteins, such as H1N1 and H3N2. There are two different options for the influenza vaccines. First, the trivalent inactivated influenza vaccine, which is injected into the muscle, and second, the live attenuated influenza vaccine, which is sprayed into the nose. Both vaccines carry 3 predicted strains yearly to be the most prevalent, based on, first, history of what's prevalent, such as H1N1 and H3N2, and the current strains across the world that are prevalent in communities. So for the United States, we would be looking at Australia. The influenza vaccine is grown in chicken embryos, which means that it contains a small amount of egg protein. Therefore, we can't vaccinate people with egg allergies. Every year, there is a new influenza vaccine that is a little bit different from the years past. So why should we get vaccinated yearly? Viral RNA is single-stranded, which is less stable than double-stranded RNA. Therefore, mutations during replication are very common, which could create slightly different daughter viruses with variation in the proteins on the structure of the virus. Therefore, even if people have been vaccinated for certain strains of the virus, those antibodies may not pair directly with these new daughter viruses, and the vaccine could be ineffective. This process is called genetic drift. The idea is that if we could predict a strain, we can make a yearly vaccine that attacks the strains most prevalent in that particular year. Because of the nature of the influenza vaccination, we aren't able to vaccinate certain individuals, such as people with egg allergies, children under 6 months old, pregnant women, and other immunocompromised people. Therefore, we activate a system called herd immunity. The idea is if we vaccinate healthy individuals, there's less spread to those without the vaccine. Therefore, those without the vaccine are more protected by the community. This idea also applies to other viruses, such as COVID-19. The technology of making vaccines is improving. During COVID-19, we learned that new stable mRNA vaccines can be produced more quickly, which improves our ability to create a vaccine that is effective against the correct strain of the flu. Other technologies are exploring how to target more stable stem regions of the virus to help our immune system identify influenza irrespective of the highly variable head proteins. A universal vaccine now seems within reach, and hopefully soon the flu's ability to infect and affect billions will become a thing of our past.